did you find out from him?
Yeah.
Do you have the link?
Yeah, but my thing is screen recording right now. Where's my iPad? Actually, let me go through this.
Austin.
Why are you giving him back shots? Where is it? Right here. Why isn't it working? Is it not going? Is this not a screen record? It is. Why is it not showing anything?
I can't wait for your hair to grow back, bro. Genuinely, I can't. Bro, I can't wait.
And so we're starting with a very basic system, the operons of E. coli, and we're starting with the lac operon. Oh, that noise again. And so we started off just introducing the lac operon in last class, and we talked about that we have mutations that were now constitutive for production of beta-galactosidase and lactose permease, two major products from the lac operon. And remember, we said that the wild-type condition was that in the absence of lactose, the operon was off. In the presence of lactone, it was on. Okay, off meaning not expressed, on meaning it is expressed. And it turns out that the mutants that were constitutive, in other words, on all the time, pointed to the fact that the operon was regulated by a repressor, a protein whose job is to keep expression off. And lactose would have the repressor get removed, okay, and allow transcription. So for a repressor to work, it has to be made as a protein and then bind to the promoter region in a sequence called the operator, okay, and that's where it binds. You can think of it as the landing pad for the operator. And the two classes of mutations disrupted that function. One was a mutation that stopped the transcription repressor from being made. Okay, those were lac I mutations. Letter I.
Yeah, that's
and so those are I minus. And then we had these constitutive operator mutations, which mutated the binding, the site sequence of DNA, so that the repressor could not recognize it. So we'll go into some more genetics of how these kinds of mutations behave in terms of dominance and recessive in a minute. But I first wanted to just kind of show you a little bit about the protein structure of how the lac repressor, what the lac repressor looks like, and its functional parts. Okay? And this will actually be kind of a good introduction to what a transcription factor what does. So this is the protein sequence in the
you know how he doesn't
tertiary structure, so the three-dimensional structure of the entire protein. But this is one monomer of it. Okay? The lac repressor actually works as a tetramer, so 4 copies of this bound together. But you can see it's got different domains, meaning different functions. Okay? There's the amino terminal region that has the DNA binding domain. Okay? That's the part of the protein that actually recognizes the bases in the DNA. So this little structure here, which we'll talk about a little later, you can see that these two alpha helices, and they actually find the appropriate base sequences in the operator. So this just doesn't bind any random sequence; it actually recognizes the operator sequence. Then at the you also then have this region here called the inducer binding domain. That's the part where lactose binds to. And when lactose binds, it causes an allosteric change that stops the DNA bindings part from working. Okay? So you could think of it as kind of the communication part, the signal. And then down on this end, you have the multimerization domain. That's important for forming the tetramer, the 4 copies together. So one of the things that you can also start to think about is that, well, if I just knock out this protein, mutate it in a way it can't function, okay, there's going to be no repressor effect. But when you have different domains, it's possible to mutate part of the gene, so one domain doesn't work, but another domain does. One domain doesn't work, but the other domain still does. So you can imagine mutations that mutate the inducer binding domain, so it will no longer recognize as lactose. What do you think would happen with that type of mutation? It will bind DNA in the crest, but it can't recognize the signal, lactose, so it's permanently stuck on the DNA. And they actually did find this kind of mutation called a super repressor mutation. It's a very rare allele, but they can find it. The problem is, it's just as we just said, it can't be removed. It's permanently stuck. Okay? So now let's get back to kind of how mutations in either the operator or the repressor itself, how they behave genetically. And so we're going to introduce kind of
your message says otherwise.
But you're going to see that there's some common things.
I don't know.
So in order to do
I just see a bunch of girls.
tests,
I was texting Asia the link.
You need 2 copies of a.
No, I wasn't.
Right?
I was
if you remember
texting.
from Mendelian genetics, right? You never could just pick one allele by itself and ask, is it dominant or recessive? You always had to compare it. So we have to be able to do a comparison in E. coli. But E. coli is a monoploid organism, so it normally only has one allele. So we need to add in a second allele to test for dominance or recessiveness. And we're going to use a plasmid to do that. And so you can see here we've added a second copy of the lac operon.
I thought you were hitting yourself
and
for a
we've got
second.
then the chromosomal copy here. And what we'll be doing is creating 2 genotypes for the operon
My
in the
water
cell.
is done.
And this will allow us to look for dominance or recessiveness. So in this example here, we have a wild-type operator and beta-galactosidase, but the lac repressor is mutant. So by itself, this will be constitutive. But what happens if we add a plasmid in with a second copy of lac I that's wild-type? Well, because it's a protein that can move around the cell, wild-type lac repressor can get produced from the plasmid and still bind the operator in the chromosome. Okay? So in this test here, LAC-I minus is recessive to LAC-I plus, because you can think of that you're heterozygous for wild-type LAC-I plus wild-type LAC-I and LAC-I minus. And when you put both together, you see the wild-type beta. The general point here is that transcription factors are proteins, and they're made by separate genes than the gene they regulate typically. And so if you provide a wild-type copy of the gene to produce that transcription factor, it can freely move around the cell and bind to whatever DNA binding sites it needs to recognize. So the key thing to remember there is that transcription factors act in trans, meaning they can move
blood
from
on
one
it?
location to the other. That's not going to be true of the binding site that the transcription factor binds to. The binding site, the operator, will only control the sequence it's adjacent to on the same sequence. So in this situation here, we have our operon in the chromosome. It makes a good LAC-I plus wild type, but it's got the constitutive operator. We can provide a second copy of the operon with a wild type operator, but because it's a DNA site, it can't leave the plasmid and regulate the chromosome. So the landing site of the operator only regulates the sequences it's next to. So the analogy I like to make here is suppose you've got a wall switch, right? And you break the wall switch. It doesn't matter how many other good wall switches you have in the building. That one is broken, so you can never come on. You can think of the
Why are
operator
you
as
starting this?
the
I will drink your water.
But suppose you also have the person who turns on the switch.
Yeah, I'm gonna drink your water.
If I'm there, I can go turn it on. But if I'm not, then you can go turn it on, right? So we're kind of the transcription factors that can move around from switch to switch,
Why is she-
operating.
I
right?
won't
So
drink
the
your
operator
water.
itself is kind of the switch
You know what? I
mechanism.
won't drink your
It has
water.
to be functional next to the gene it regulates.
Asia.
Transcription factors can work from different locations to move from operator to operator. So it's not unusual for a transcription factor to be regulating multiple genes, not just one particular target. And so we say that the operator is a cis-acting site, meaning adjacent. Okay? So all promoter sequences, right?
I think
Octobox,
that- I think you
those
should.
are all cis-acting, okay? Because they only, if you mutate
I
them,
can
it only
see you
affects the
trying
gene
to jump
that they
on
mediate
things
transcription.
and
And finally, let's just take a look at our super repressor. Well, super repressor is the version of lac repressor that can't recognize the lactose. So once it binds to DNA, it's kind of stuck there. It's going to behave dominantly to wild type lac repressor because you can see that if you're producing two copies of the protein in the cell, a wild type here and a mutant here, the mutant is going to be able to go to this operator in the chromosome once it binds, it can come off. So this is kind of a gain of function mutation.
I'm
So it's going to be
sorry.
dominant. So it's
Sorry.
like hypermorph, if you remember that word.
I just did.
So this slide here kind of summarizes the different mutations, and this can be useful when you're doing the homework assignment for next week. Okay, it's assigned now. Also, I have a video that I just posted up, kind of working through how to solve the operon genetics problem. So the typical problem you're going to get is you're going to be given genotypes for a cell that has two copies of the operon with different allele combinations of them, and you're going to have to come up with the overall phenotype of the cell. You know, is beta-galactosidase based? Is like permease based. And when you look at the video, you'll see that I kind of have a systematic way, I say, to assess each gene. You know, you'll want to start with asking, is the part of the lac operon that encodes the enzyme, the part that encodes LAC-Z, or the part that encodes LAC-Y, is that mutant? If that's mutant, well, you're never going to get activity. So then you look at the operator. You ask, what does the operator look like? What does the promoter look like? So you look at the cis-acting sequences for that gene. If those are all good, then you look at lac repressor, the transcription factor, and see how that is.
Where do I have to go over here for? The water's here.
So I wanted to get back to the idea of how transcription factors bind DNA and really kind of emphasize kind of how using proteins like lac repressor, the specificity of transcription factors for recognizing certain sequences was figured out. Okay? So you see the structure right here. Here's the DNA binding
was so
domain,
good.
and here's that helix turn helix, these two alpha helices fitting into the major grooves of the
What are you
DNA,
doing?
and it's able to purposely pick out the operator sequence. So how did researchers first figure out
Yo,
that
she's gonna break her ass.
repressor actually bound to DNA? Well, they did a type
Nick,
of experiment
my
that's called DNA
I'll take like one.
and I'm going to walk through various parts of this experiment to show you
It's in your
the top part.
You touched the Amazon with that, by the way.
So
When you touch
what we're going to do is we're going to do two experiments in parallel where we have DNA sequence containing the operator. Okay? So that's what this picture here is. These are the DNA sequence with the operator. And what they've done is they put a radioactive phosphate on the end. That's pretty easy to do in the lab. And then half of it they mix with repressor protein, and the other half they leave unbound. So they've broken the experiment into two parts. Labeled operator,
Why'd you touch
bound
my
repressor,
gummies with
labeled
your
operator.
dirty hand?
That bitch.
And then what they're going to do is they're going to take these two samples and digest them with an enzyme called DNase that cleaves DNA in specific spots, kind of like restriction enzyme nuclease. Well, what's going to happen here is if you have repressor bound, the repressor protein is going to block the nuclease, the DNase, from getting there. So it won't cut the sites where the DNA is bound. So if you then take the digested products with the protein and without the protein and electrophorease them on a gel, you'll see missing bands where the repressor protein was bound, that would block the sites. So without repressor, you would be able to cleave it at all these different sites. When you have repressor brown, you can see that there were some missing size fragments, meaning you never were able to cut the sites where the operator So they call this kind of DNA footprinting, because you're looking to see where the DNA is basically stepping on the DNA. No, sorry, the repressor is kind of stepping on the DNA and blocking it. And if you combine this with DNA sequencing, you could actually figure out exactly what bases were footprinted. Okay? And so this is how experiments are still done. There are better techniques for this, but the idea of being able to identify exactly what sequences a transcription factor is bound to is still a method that's used today. Okay? What this also told researchers that lac repressor just didn't randomly bind DNA. It actually found a specific location and stuck there. So if we look at the promoter region sequence of the lac operon, we can actually map out which proteins are bound to different parts. So just for reference, this is the ATG, the first amino acid found in beta-galactosidase. Here's where the mRNA starts, and then going back a little, this is actually supposed to be a continuous line starting from the top left to the lower right, so this is one long piece of DNA. The brackets indicate which proteins are sitting on which parts of the sequence. So here's the operator sequence, and the repressor protein is bound in this region. So you can see that it basically sits right where transcription should initiate. RNA polymerase footprints a much larger region, starting all the way from here and then going all the way to here. What do you notice though? The operator site and where RNA polymerase would bind overlap. So you can imagine that if you have lac repressor sitting on the operator, RNA polymerase can't get there. So it's really kind of a competition. You'll also see that they've indicated another site here called cyclic AMP binding site. We'll talk about that a little later. Right? I just wanted to show you here is that researchers have actually been able to try to crystallize the lac repressor with DNA wrapped in it. And they actually find that it has the primary operator site that we see here. But there's actually a second operator site about 100 bases away, and it actually causes the DNA to basically form this big loop with the repressor protein in the middle, the lac repressor protein over here. So this is actually the heterotetramer of lac repressor. Two copies here, 2 copies here. And it's binding the 2 operator sites. Something you may see here, if you look closely at this region, it looks like 2 hands grabbing up at the DNA. Okay? Each little hand is that DNA binding domain. So this is one copy of the DNA binding domain, here's another copy. And what they're binding to is the sequence right here. If you look at the operator sequence, you'll see that it's palindromic. So the sequences on this side are kind of similar, mirror images of the sequences on this side. That's what the 2 hands are grabbing. Okay? And then imagine there's another one of these sequences about 100 bases away, up in this area. And so now you've got 4 hands grabbing. And that's kind of what the picture you see here is. So, on that previous slide, it mentioned something about cyclic AMP. Well, there's part of the story I didn't tell you yet. We talked about lactose. So here's an experiment. If we have our E. coli and we have no sugar present, there's no expression of the lac operon. Also, the E. coli won't live too long because they actually need a little sugar. If we had glucose, the primary source of carbon, the favorite, there's no expression of the lac operon because why waste trying to make lactose metabolizing enzymes if you're getting the best carbon there is, glucose? Now let's say we switch it around and we take away the glucose and we go to lactose. Now what happens? Expression comes on. So just like Jacob and Monod saw, that when you switch from glucose to lactose, expression goes from off to on. But what happens when you add both sugars together? Okay? Well, what you see here is that it's off again. So what does that tell you about the regulation? Okay? So now we have 2 sugars coming in here. Well, it starts to tell you that the glucose is not just outside the regulation, it actually has a role. The presence of glucose actually turns off the operator. So this is a general concept found in lots of organisms, that because glucose is the favorite carbon source, it represses promoters of other sugar utilization pathways. So the lac operon is controlled by both sugars. The presence of lactose turns on the expression. The presence of glucose keeps it off. And when you have both together, the repression wins. So how does that glucose repression work?
Give me my dick.
Well, it turns out that
Enough of you
the
guys.
transcription factor here
You guys don't
doesn't
wanna stay,
recognize
you
glucose
guys
directly.
don't wanna
It recognizes cyclic AMP. And it turns out that when you're starved for glucose, your amount of cyclic AMP goes up. There's a specific protein called CRP, catabolite repression protein, and CRP will bind the promoter when cyclic AMP is present. So here's the CRP monomers existing unbound. Okay? And so with high glucose levels, CRP is unbound. You starve for glucose, cyclic AMP gets produced, it causes the CRP monomers to dimerize, and then they bind DNA. So, in order to get expression of the operon, we have to add the lactose, which removes the repressor, but starve for glucose so the activator CRP binds. So the lac operon is dually regulated two different mechanisms. Lactose is needed to take the repressor off. Glucose is needed to make starvation is needed to activate it. Okay? So you can see here then there's 2 different signaling systems feeding into the regulation. And this catabolite repression protein actually regulates many different genes. Because you can imagine that glucose starvation is going to be needed to turn lots of different pathways together. You also see here another theme is that the binding of a signal molecule, say cyclic AMP in this case, to a protein causes a structural change, which we call an allosteric change. So yeah, that's what you see happens here. The monomers dimerize and now can bind DNA. Okay? And so this ability of a small molecule, whether it's lactose or cyclic AMP, to alter the structure of a protein so it binds or doesn't bind DNA is a very common thing found all through life, this allosteric change engine. So if we go back to this picture here, okay, well, no, I'm throwing this, here's the DNA sequence I showed you before. This is what this promoter actually looks like now. Okay? You've got the operator, RNA polymerase binding, and then cyclic CRP, metabolite repression protein, binding adjacent to RNA polymerase. And the thinking is that RNA polymerase needs some help getting started, and that's what CRP is kind of doing. It's making basically it makes RNA polymerase work better to drive transcription.
Spilled it on me with the
There are some people who can say that everything you need to know about how transcription work was found with the lac operon. I mean, it definitely lays the groundwork for themes that we'll see in lots of different gene transcriptional regulation stories. So now we're going to switch to a biosynthetic pathway where we're going to see feedback inhibition, of the pathway by the end product. So remember we said that there were these two kinds of systems, inducible systems. So lac operon is an inducible system because the presence of lactose induces expression. We also have these repressible systems where the end product of the pathway turns off expression, typically seen in amino acid or nucleotide pathways, where if you can get the tryptophan for free in this example, you don't need to try to produce it yourself. It's energetically quite expensive. So we're going to take a look at the operon for regulating tryptophan biosynthesis, and we're going to introduce a new mechanism of regulation that's not going to rely on a transcription factor, but instead is going to rely on RNA structural changes and RNA sequences that regulate expression. And finally, we'll end this part of the lecture with talking about how this really thorough understanding of the lac operon actually has uses outside of E. coli biology.
Yeah. Yeah.
So
This is
let's
what
look at the
I
trip
was
operon.
living with.
So as I mentioned,
That we-
most
if I was to stay
genes
in
in
Boston.
bacteria actually are arranged as operons, where you have one message and multiple proteins translated from that message. And it's thought of that evolved that way so you can regulate genes in a common pathway by just regulating one transcript, right? So you want to metabolize lactose, you need the permease and the beta-glucosidase. Well, you put them both on one RNA, and so you just have to regulate transcription of that one message. For tryptophan biosynthesis, it turns out there are 5 enzymes needed to make tryptophan, and so we're going to make one mRNA that encodes all 5 of them. Okay, remember,
Only
5 separate
30
trideltarnos
minutes
that
in?
initiate translation for each of them. So here's the structure of the trip operon. You can see some general commonalities, right? Here's where the message starts over here. And
Y'all
these
have
letter
issues.
gene
I
names
hope
A, B,
y'all
C,
know.
D, E are the 5 enzymes that make tryptophan.
I just wanted to.
You may remember that
food
TRPA was the
and
enzyme that
peas.
Charles Janowski was using in that experiment where he mapped the mutations and showed that they affected the position of the amino acid in the protein. So at any rate, the initial characterization of this operon suggested that it was regulated by a repressor protein called TRPR, okay? And TRPR's job was to bind to the operator, and it bound when tryptophan was present. So the signaling molecule is tryptophan, the allosteric changes, tryptophan, the amino acid binds to repressor, causes it to bind DNA. When you're starved for tryptophan, the repressor is unable to bind DNA, so it comes off. So you may think that what would the phenotype be of a mutation that knocked out production of TRPR,
Where's
right? No
my-
repressor. So that should be on even when tryptophan is present, a constitutive mutation. So common theme, repressors normally keep expression off. Mutating repressors cause expression to come on under conditions where it should be repressed.
Who cares?
So,
No.
Researchers thought this was the whole story of the trip operon, but we're going to see that it's a little more complicated.
You can get more.
What researchers noticed was that there were 2 different size messages in length made by the trip operon. There was a longer transcript that would actually encode the proteins for tryptophan synthesis, and then this shorter one. And they noticed that when tryptophan levels were high, you made the shorter transcript. When tryptophan levels were low, or complete starvation, you made the longer one. So they started to reason that somehow tryptophan concentrations in the cell was somehow deciding whether you make the long or the short message. And remember, you'd want to make the long message when you were totally starved, because that's when you want to make tryptophan. You'd make the short message, okay, when you had a little bit of tryptophan around. Okay? And then on top of that, you've also got the repressor protein binding here. So this becomes a little clearer when we look at this table. So what we're looking at here is the amount of expression from the trip operon, okay? And without tryptophan, the starvation condition, that's 100% expression. That's the maximum. In wild type, when you add tryptophan, you go down to about 8% of that 100, okay? So it's not 0, okay? But it's not 100% either, right? You're from 100 down to 8%. So you would hypothesize, based on what you know from lac operon, that if you mutated TRPR, this value should go all the way up to 100, right? Because if the repressor is removed, there should be no other way to repress. It should go up to 100. But that's not what they found. Instead, it was up a little, but still a third of what was found with starvation. So this told researchers that there must be a second repression mechanism that senses tryptophan. Okay? And that second mechanism is going to be this regulation of RNA length. So I'll tell you the big answer now, and then we'll look at the details. When you have full levels of tryptophan present, okay, you have the repressor protein bound, and there's no transcription. As you start to starve, the repressor protein comes on, but it'll first only make that short transcript. Okay? It's not until you're completely starved for tryptophan that you make the long one. So if we go back to this picture here, okay, you've got full repression with the repressor protein, partial repression, and you make the short message. No repression, you make the long message. Okay? And there's going to be this correlation with the presence of tryptophan. And when I talk about starvation of tryptophan, we're actually talking about sensing the charged tRNAs with carrying the tryptophan. The charged tRNAs for the tryptophan codon. Okay? So, because where is the tryptophan going to be used? Translation. So we need to therefore tie in translation to transcription using tryptophan as a monitor. You know, it's a lot of transcription translation, right? It's getting a little confusing. And this is not an easy concept, okay? So you should be a little confused with this. I have to kind of review this every time I go through it. Okay? But what we want to do is figure out how does decreased tryptophan cause you to switch from a short message to a long message. And we're going to do that by monitoring translation. So how do we do that? Well, I first want to introduce the top panel here. Okay? And it turns out that there's a sequence in the RNA for the trip operon that has two structures that can go between. And these are two different stem loop structures. Remember, a stem loop is when a sequence in RNA or single-stranded RNA or DNA base pairs back on itself, kind of like the structures of, you have three stem loops in a tRNA, for example. So in this sequence, you can form either two adjacent loops, formed by base pairing 1 to 2 and region 3 to 4, or you can base pair 2 and 3 and form this one loop. And it turns out these loops have different functions. If you form the 1, 2, and then 3, 4 loop, 3, 4 is a transcriptional terminator, and transcription will stop right here. If, however, you form the 2, 3 loop, you allow transcription to occur. Okay? So we've added a little information. There's something about the sequence in the RNA that can form two structures, and they're exclusive of each other, because we're talking about one RNA, right? And you can kind of see here that those regions, 1, 2, 3, 4, okay, have two and 3 have two different pairing possibilities. 2 can base pair with 1, or 2 can base pair with 3. Okay? 3 can base pair with 4, or 3 can base pair with 2. So it's an either or. Now, where is this located? Well, this sequence is right here in this leader region. Okay? It's right around where the short and long message switch. So if you get the termination signal here, you make the short message. If you make the 2-3 anti-terminator loop, as it's called, you make the long message, which is shown here. So we now see functionally how the message can form two structures, but we still don't know the how. Okay? How does tryptophan levels change which structure you get? Well, this is where the ribosome comes in. So you may remember that in prokaryotes, we say that translation and transcription are coupled, meaning that as soon as a message starts to get made, once the Shine-Delgarno sequence appears, a ribosome goes down. And so as this message is getting made, the first part of the message is right over here where it says leader. Well, it turns out the leader has a Shine-Delgarno sequence in it for a short peptide. And that short peptide, called the leader peptide, has two adjacent tryptophan codons in it. And so here's those tryptophan codons. And if there's enough charged tryptophan tRNAs present in the cell, the ribosome will cruise right through that region, making the small peptide, because it's got tRNA carrying tryptophan. And when it does that, that forces the 3-4 termination loop to form. So tryptophan present allows translation of the leader peptide to reach through the trip codons, forming the 3-4 termination sequence. What happens if you're starved for tryptophan? Well, the ribosome binds at the Shine-Delgarno, and then it gets to those tryptophan codons, but there's no charged tryptophan tRNA. So the ribosome stalls. It's sitting there waiting for charged tRNAs, the tryptophan. By stalling, it forces the 2-3 loop to form instead, and then that allows transcription to continue. Okay? So what did we just see here? The ribosome's ability to translate tryptophan codons decides whether you form the termination loop or no termination. Okay? And remember, in the absence of tryptophan, that's when we want to stall and have the transcription proceed. Okay? So going back to this slide, what will happen is, when you have zero tryptophan around, okay, the leader peptide gets translated up to the tryptophan codons, but the ribosome stops, and you get the full message. Okay. If there is tryptophan present, you're able to translate the entire leader peptide. That causes termination of transcription. You get the shorter message. So the thinking of this is, well, how could something like this have evolved, right? So that for high levels of tryptophan in the cell, you've got the repressor protein. But then because tryptophan is a very energy expensive, I don't have the citation, people call tryptophan the most expensive amino acid to make energy-wise. That what you need to do then is basically account for every tryptophan molecule you have. And you have this more subtle regulation method that can say, okay, well, there's still a few charged tRNAs carrying tryptophan, so we'll make the leader peptide and we'll make the short message. But once I've got none, okay, now I need to get my tryptophan in there. You may remember a few weeks ago when we were talking about BLAST, tryptophan is actually a very rare amino acid. Okay? And so the cell might be able to get by with just a few charged tryptophan tRNAs in the cell. So it's really trying to look between, a few dozen tryptophan molecules and zero.
Don't touch my shit.
So when researchers first discovered it, they were trying to like,
No,
they said,
Sidon, put my iPad down.
We wanted to always
That's
believe
not
that
my
transcription
problem,
regulation
Archie.
was by protein
I'm
binding
not
to
involved
DNA.
in this.
But this now
is you
introduced the
and
idea that
Asia.
somehow RNA structures could regulate. So two things have been found out. One is that other amino acid operons in bacteria are regulated through a similar mechanism, which is called attenuation. So if you look at the histidine operon for histidine synthesis, you see that it had a leader peptide with, I think, 5 or 6 histidine codons in a row. So that's the sensor. Okay? In the same way, if you had enough charged
Put my
histidine
iPad
tRNAs, you translate through it and stop the message. If you were starved for histidine, you would get stalled in the histidine codons, okay, and then allow a longer transport. Okay? But that's still, bacteria, right? Well, researchers started to realize that RNAs themselves can be regulatory. So in next class, we'll hear about microRNAs and other types of RNA molecules that have regulatory effects. And now I'll show you a couple of other examples from bacteria where different RNA folding structures can directly regulate gene expression. So we're going to go through various versions here. And this is, we won't have that many examples. I have one example, but this is more just to see all the formal possibilities. And the theme you're going to see is we're going to have alternative RNA structures, and they are either going to inhibit transcription or translation. And a lot of times we'll see that there's some kind of signal molecule that causes the RNA structure to change from one to another. So one type of mechanism is called a ribose switch. Right? And we're going to see two examples here that are called cis-acting, meaning that they regulate just the gene they're attached to. And so, in this top example here, in the example on the left, we're going to be regulating transcription elongation. So this is going to be this is going to be the general mechanism that we just saw with the tripopyrin. So we're going to alternate between a anti-terminator versus a terminator sequence. But in this particular example, it's not the ribosome involved, it's actually the effector molecule, which is whatever the usually involved in its pathway. The effector molecule causes the alternative structures to form. So without an effector molecule, you get transcription continuing. With the effector molecule, you get this alternative structure, which causes a termination. In a different mechanism, the ribose switch folding hides the Shine-Dalgarno or ribosome binding sequence. So you can see here that without the effector molecule, the Shine-Dalgarno sequence is available for translation. In the bottom mechanism, we see that the folding up has occurred with the effector molecule, and the ribosome binding site, the Shine-Dalgarno sequence, is blocked. Okay? So the mechanism on the left is affecting transcriptional regulation, lengthening, and the mechanism on the right is affecting translatability of a message, by blocking the ribosome site. Okay? But the main theme you want to get from both of these mechanisms is the RNA can take on two different structures, one allowing either transcription or translation to continue, one not. Okay? And what changes the structure is some kind of effector molecule. So as an example, there's a methionine producing pathway in Bacillus subtilis that wants to regulate the pathway of S-adenosylmethionine. Okay?
Oh my God, it's like
regulated by S-adenosylmethionine, the precursor of methionine. So there are 11 genes that have what's called an S-box ribose switch. Okay? And the S-box ribose switch inhibits transcription when it's bound to S-adenosylmethionine. So it's kind of like this message we see example.
Yo, you're a
think of the blue
sellout,
effector
bro.
as
You
sent
fucking let Asia get the water
when it's
poured on
present,
her
you terminate
justice.
transcription. So it turns out there are 11 genes
Yeah,
that
but
all have the
now
same
you
kind of
water
ribose
on
switch
you.
sequence. That's all.
Everyone
that stops
did not
transcription
benefit from
when
the
SAM is
situation
present.
but her.
So that's kind of the ribose switch.
Because she has
We
water
can
on
also
her
have
mind, you don't.
RNAs called
So it's like-
short RNAs, okay, or small RNAs, that can work in trans to block other genes from getting translated. So in this particular example here, okay, the transacting small RNA is complementary to the gene's ribosome binding site, and so when it binds the RNA's when it binds the ribosome binding site, it blocks translation. Okay? So this means that you have to have a separate locus in your cell that produces the small RNA, and then that would be regulated to come on when you want to turn on translation of the target gene. Okay? So remember, the small RNAs basically are targeting another gene's message to inhibit translation. In this example here on the bottom, the small RNA is letting translation occur by changing the structure to free up the ribosome binding site. So these are just two different mechanisms where one, the small RNA acts as negatively to turn off translation. The other example on the bottom, it acts positively to allow translation to occur.
I could have went upstairs.
Researchers have figured out to design these small RNAs artificially to turn off particular genes by blocking the ribosome binding site.
Fuck no.
And then finally, you can even design what are called antisense messages by expressing the entire or a portion of the antisense mRNA complementary to the mRNA. And what would happen is if you put in enough of the antisense RNA, which is the blue, it would bind up the message and stop it from getting translated. Again, you would have to somehow provide a source of producing the antisense message. Remember, the sense version is what's translated. Antisense is complementary. There are some actual examples of particular genes that actually have evolved to have an antisense message made to purposely turn it off. But a lot of times this mechanism is now employed in experimental situations where you try to target inhibit translation by making an antisense mRNA.
What's the point of all this?
We're going to see that something like this mechanism works in eukaryotic gene expression as well. Now, finally, I wanted to get back to some uses of the lac operon. As I mentioned, it's kind of the first and probably one of the most studied transcriptional regulation systems. And if you think about it, you've got this promoter that you can keep off in the presence of lactose and then allowed to come on when you're starved for lactose. You've also got this enzyme, beta-galactosidase, which can metabolize lactose, but also can metabolize synthetic sugars. And there's one synthetic sugar that actually will turn blue when it's cleaved. So this is the first really use of a protein called a reporter. How many of you have heard of the term of kind of a gene expression reporter? Anybody of you know another example of a reporter?
Monkey
Protein.
paws touch it?
Yes, monkey paws touched it.
Right, that's you have a reporter that way. And then anyone know about, ever hear of a fluorescent something called green fluorescent protein? That's another reporter. So a reporter is just a general term to mean a protein that gives a visible readout when it's active. Okay? Just think about it, most proteins are invisible to us in the whole organism. You have to use some kind of Western blot or fluorescence imaging to see the protein. But with a reporter, you can assay the cells and see expression. We'll see green fluorescence protein in a few minutes, but here's just an example where we've taken the beta-galactosidase protein and expressed it on a different promoter in E. coli, and it will turn blue. You could actually take this gene and put it into mice and plants, and also see blue color where the enzyme is active. So remember, when we use it as a reporter, its transcription is not being driven by the lac promoter. It's driven by some other promoter that you're wanting to test. Okay? And we're just using the LAC-Z activity, the beta-galactosidase, to report on that gene's expression.
You can't be like, we should make a mirror.
The other, so that's a lot of basic research involves using it as a reporter. The other tool from the lac operon is the promoter. Right? So you've got a system where you can keep a gene expression off with glucose and then on with lactose. Well, a lot of research in biotech is designed to take a gene or protein for, let's say, a mammalian peptide hormone or something like insulin, and then produce a lot of it in E. coli as a factory. Okay? Well, if you try to produce proteins in E. coli, many of them are toxic if they're produced at high levels, meaning they'll kill E. coli. So what researchers want to do is grow up E. coli, so they got a lot of cells, but keep the protein that you want to express from getting made until near the end of the experiment. So if you take your protein of interest, let's say, mammalian insulin gene, you would put it into a plasma that has the lac promoter driving it, turning it on. You grow up those cells in the presence of glucose, no insulin is made. Then you shift them to lactose medium for a few hours, and then all of a sudden you can get a lot of protein. And then the cells will probably start to get sick, but by that point you actually take your cells and lyse them and purify your insulin. So using one of these type of expression systems, you can sometimes get to like 1% of the total E. coli protein is the protein you're trying to produce. Sometimes even. But so this idea of using the lac promoter as a regulator when you choose to produce some foreign protein in E. coli is also something that's commonly used. Okay. So there's the sales pitch for E. coli and lac operon. But now what we're going to do is move from the general themes of transcription from prokaryotes and move to eukaryotic gene regulation.
And he runs his mouth too much too.
So first point is that we're going to see that gene expression is regulated using many of the same themes that we see in prokaryotes, but more complex, meaning more mechanisms for regulating expression and a lot of overlapping regulation. We're going to see that we have cis-acting sequences that mediate transcriptional regulation, and these cis-acting sequences are referred to as enhancers. So enhancers are sequenced in the DNA near the promoter region, and that's where transcription factors are going to bind to the enhancer. We'll see an example of that. And then we'll also see that another layer of regulation comes in with the chromatin structure of our DNA. And if you remember, we have the idea of heterochromatin generally describing regions of DNA that aren't transcribed or transcribed, and then euchromatin where it is transcribed. And what we see is that there are ways of basically protecting one enhancer from another in nearby genes with sequences that are called insulators, for example. So here's kind of our general overview of eukaryotic gene expression. It's kind of familiar. So we've got our genomic sequence. Okay? We get a primary transcript where we will remove introns if we need to. We will cap the message and we will polyadenylate it, and then that will be moving it out of the nucleus for translation. Okay? So we're not going to have this coupled transcription translation in eukaryotes that we see in prokaryotes. And then our protein is going to get made, and we can see some modification to the protein. Okay? So we also want to, for now, think about transcriptional regulation is the primary way we regulate how much protein we end up producing, right? Because if you don't have the message made, you can't translate. Okay? And still today, while people do study mechanisms on regulation of protein activity, okay, the primary way you can change how much of a gene activity you have is by changing the level of message. Okay?
Who's the other girl? What other girl then? Sing with the other girl's name.
Let's look at what we classify as a promoter and its other cis-regulatory sequences in a typical eukaryotic gene. So we're going to have start here at transcriptional start site, and we always call the first base that gets transcribed in the mRNA +1, and then the bases that become the message go up positively from +1. Going the opposite direction, we go in the minus direction, and those are going to be the sequences that contain the promoter and the enhancer. The promoter being the region where RNA polymerase binds, the enhancer being a sequence where other transcription factors bind. So first thing we want to see is that the promoter sequence where RNA polymerase binds does have to be physically very close to where RNA transcription binds. So we're going to see that typically within -25 to -50 region is where RNA polymerase is going to bind. And there's a sequence kind of equivalent to the sigma to the -10 box called the TA sequence. We'll talk about that in a second. But then the sequence where our transcription factor binds the enhancer doesn't have to be right next door to the promoter. It can be thousands of bases away in the minus direction, but it can also be located even on the 3' end after the coding sequence. So this is the second choice here. The enhancer is on the right, enhancer is on the left. And these are just generalizations of what's been found in typical genes. So a lot of work is done trying to identify enhancer sequences, because remember we said the enhancer is where specific transcription factors bind. They are going to give specificity to when a gene gets expressed. I'll mention this now, is that, and we'll talk about it on the slides, that you can think of the promoter and RNA polymerase alone providing a low level or basal level of transcription, and then the enhancer mediating a transcriptional activator to come in and express at higher levels. So we're going to see that there are enhancers for genes that direct expression at different developmental ages or in different tissues. Okay? So they give the specificity. And in this example here, what they're showing you is how we can use one green fluorescent protein, this other kind of reporter, okay, to see how active an enhancer is and the importance of it. So in this case here, they've taken the wild type sequence of the gene, separated it from the gene encodes, and used it to drive expression of green fluorescent protein reporter. So if we leave the enhancer and promoter intact, we get a high level of expression, and we see that the expression is in the eyes. However, if we use a mutated version of this that only has the promoter but no enhancer, the expression is very low, or what's called basal expression. So this is the main point. The enhancer adds the tissue specificity and the higher level expression. Here's another example of a reporter. This is actually a small worm called C. elegans, and this is a gene that's expressed in the what are called pharyngeal neurons of the mouth. Okay, and so its mouth end is down here, and these two big green bands here, bright green, are two very long neurons that are involved in feeding. And this happens to be a promoter for a gene that's expressed in the
That would be
pharyngeal neurons.
cool, yeah.
This is probably less than a millimeter long. This worm, so this is really close in zoom.
A sports medicine club?
So let's talk a little bit more detail about what these transcription factors do. Here they are binding to the enhancer, and what they can do is cause the RNA polymerase to either bind better to the promoter or to be more active. Okay? We'll talk about some mechanisms of how that is in a few minutes. But here's kind of the two main parts. There's usually more than one enhancer and more than one transcription factor binding on the regions. At the promoter,
But like
we're going to
there
have
was
RNA
no
polymerase.
like it
RNA polymerase is a lot more complex in terms of subunits and proteins that are involved. And so this next slide is showing you just RNA polymerase binding at the promoter and large complexes of protein that are involved. Okay? So the TBP or TATA binding protein is the equivalent of sigma factor. Its job is to kind of bring RNA polymerase and accessory proteins to the promoter. So if your gene has a mutant TATA box, RNA polymerase is not going to bind very well if you delete it. Okay? So it's a cis-acting sequence. Now, the other proteins involved in here are not only RNA polymerase 2, which is more than a dozen subunits, but also these what are called TATA-associated factors or TAPs, which kind of help this interaction. So let's just think about this being about 25 to 30 proteins. But because we're kind of geneticists, we're just going to draw it as a few colored blobs, okay? But it's quite a large complex of proteins, and you can see that it's bound in the region of about -30, and here's the start of transcription over here. Okay? So let's go through some examples of how a eukaryotic transcription factor will work. First, we're going to look at activators, okay? These are proteins whose job is to increase transcription from a gene, make RNA polymerase work better. Here's kind of your classic example where the activator protein is bound to both the enhancer and contacting RNA polymerase physically. And you can see that this is why the distance between the enhancer and the promoter is not that important, because the DNA can loop around so the proteins contact each other. There are also examples where activator proteins don't interact directly with RNA polymerase, but instead they change the chromatin structure to make the
I thought you saw
to
me.
change the nucleosome structure so the chromatin is more accessible to RNA polymerase. But this then brings us to kind of some general themes about the structure of a transcriptional activator. It's minimally got to do two things. It's got to bind DNA in a specific way, kind of like the LAC repressor does, right? So they have a domain called the DNA binding domain. And then it usually has what we call an activation domain, which is usually involved in either contacting RNA polymerase directly or working to change the chromatin structure. And these are usually separable domains in that you can just express the DNA binding domain by itself or the activation domain by itself, and you can assay one activity instead of the other. You can also make synthetic transcription factors by mixing together DNA binding domains and activation domains from different transcription factors to get new activities. Many transcription factors, in fact, pretty much all of them, usually function as multimers, typically dimers, so you also have to have a domain for forming the multimer. Okay? So, as it says here, minimally 2, but usually you have this third dimerization domain. Now, when we talk about DNA binding domains, we're talking about structures that have evolved to read specific sequences within the DNA helix. There's a small number of domains that have evolved to do this, and so we see them show up. Structurally similar transcription factors show up all throughout life. Okay? So you may remember we talked about the LAC repressor having these two alpha helices. Okay? Well, this structure called helix turn helix shows up in both eukaryotic and prokaryotic transcription factors. So once you figure out a good way to bind DNA, it keeps getting used over and over again. Okay? Amino acid changes will specify what sequences in the DNA it can read, but the general theme is still present. Here's SP1. We talked about that having those cysteine residues, and the zinc fingers, or what the structure is called, are found in all throughout eukaryotes as being kind of a good way to recognize an enhancer sequence. Now, I said that transcription factors usually work as dimers. They can work as homodimers, meaning 2 copies of the same protein, or sometimes they'll mix and match to make heterodimers, which will give you new specificities. So in this example here, we're looking at a transcription factor family that has 2 proteins in it. One is called Jun, J-U-N, the other is called Fas. It turns out that Jun homodimers bind one particular enhancer sequence. June phos heterodimers bind a different enhancer sequence. Apparently there's no phos-phos homodimer. And so you can imagine by altering the ratio of June and phos, you can have a particular cell have more of the heterodimers or more of the homodimers. And that would then help figure out what types of genes you're going to be expressing.
No, you won't.
I'm
Now,
gonna bet.
repression in eukaryotes also happens, so it's not just activators. And there's various different mechanisms that we can think of to do this. One is kind of analogous to the activator, except that, so you bind your enhancer, but then you inhibit RNA polymerase from binding through what's called the co-repressor domain. You can also cause the promoter to be put into denser chromatin, heterochromatin, and in this case, that's what the repressor protein is doing here. There are examples where the same transcription factor can, in one scenario, be an activator, in a different scenario, be a repressor. So in this example here, the dorsal protein working on its own is a transcriptional activator. So it's got the DNA binding and activation domain, the two domains. Okay? But if it's present with a third protein, second protein shown in gray, that then brings in a protein called Groucho. You can tell this is Drosophila protein, right? So it's got like a semi-humorous name. Anyway, Groucho comes in and binds, instead works as a repressor. So the common thing in here is that dorsal, depending on the cellular context, can be an activator or a repressor. This is kind of just a thematic slide showing you the different mechanisms for repression. In one example, you can have competition for the same enhancer sequence. One protein is the orange protein is a repressor and blocks the activator from it. So just competition for a site. You also can have examples where the repressor protein binds directly to the activator, stopping the activation domain from working. That's called quenching. So you can imagine one cell type where you have the activator protein, the quenching protein present, and another cell type where it's not. So off in one condition, on in the other. We also see examples where we can have transcriptional activators that are retained in the cytoplasm and only after a particular signal get to go into the nucleus. So the transcription factor involved in inflammation and immune response called NF-kappa B binds DNA to turn on genes, but if the inhibitor protein I-kappa B is present, it keeps NF-kappa B out of the nucleus and in the cytoplasm. When an appropriate signal comes, the inhibitor is degraded and the activator goes into the cell and turns it on. So sequestration. And then finally, different heterodimer, homodimer complexes can be activator or repressor. Again, no specific examples here, just general themes. We even have some transcription factors that, kind of mechanistically work somewhat like lac repressor, okay, or CRP. They are bound by a signaling molecule, and that changes the structure to be able to bind DNA. A classic example of this are what are called the steroid hormone receptors, okay? The steroid hormone gets into the cell, it binds to the transcription factor in the cytoplasm, that then causes it to go into the nuclease and activate transcription. The binding of the steroid causes an allosteric change in the protein to make it be able to bind DNA. So just a couple of more slides for this part. So this is kind of a summary slide showing you how different combinations of transcription factors repressors can regulate a gene, both in terms of which tissue it's expressed in, spatial expression, and also when in development or temporal changes a gene is expressed. Okay? So you can see the same gene here getting expressed under different conditions depending on which different transcription factors are present. Of course, then this creates, there's always a step back, this is our target gene we want to express, but now we have to figure out what decides which transcription factors and repressors are made, and that becomes then the earlier step in regulation, right? So in other words, our target gene depends on when these other regulators are expressed. And just the last case here, bringing in the reporters again, we used the reporter as an example to study enhancer sequences. We can also test the function of different transcription factors by having a wild type enhancer, but having mutants in the different transcription factors. So, you can test the functionality of this yellow transcription factor by mutating it, and you see expression goes down. Okay? So we will stop there and then finish off this chapter in the next class.
So this method was kind of great for a while, but there's a couple of problems here. Okay? One is it takes several days to do this experiment. Okay? The other is you can only look at the expression of one gene at a time. So every gene has to be looked at individually. So researchers then developed what's called a PCR-based method, called inverse transcription PCR. Okay? And the principle here is, again, we're going to have to look at one gene at a time, but we're going to be able to get better quantitation and it's faster. And what we're going to do is we're going to take our RNA, convert it all to cDNA at basically a 1-to-1 proportion, and then we're going to do PCR with gene-specific primers for a particular gene.
Why did you do that, bro?
Okay, we're going to quantify how much
Someone's
PCR
like
product
2
is present
pounds
in every
soaking wet,
cycle
but she will
of
still
PCR.
stop me from doing
Can
anything.
go on for maybe 30 to 40
Do
cycles
the look
of
you just gave me.
nature synthesize.
You want to do it
Into
too?
the product. And it increases exponentially. So if I start with only one copy of the cDNA in my
She just don't touch me.
it might take me 30 cycles until I see a product. But if I started with 100 copies of the cDNA, I should get product a lot faster, maybe at 22 cycles, 23 cycles. And so if I can measure
Maybe
at which
want
cycle
to.
my PCR product appears,
It's so
that tells
good.
me
It's
relatively
actually
how much
so
time
funny,
was present,
bro.
how much cDNA was present,
So
and
it
how much time.
stares,
So
Asia
the
just
earlier
doesn't care,
cycle
and
my product comes up, the more abundant.
she gets the
So this is a
reaction
great
like
way for doing
once.
quantitation and measurements.
See?
And because you can do things with much more accuracy, you can get easily
I know, I understand the play now. You just
1.5-fold
wasting
changes in
time.
expression. So better for speed, better for accuracy, Okay? But still, you're doing one gene at a time. You know, you can, yes, in PCR experiments, you can maybe do, 10 genes in one experiment. That's still a lot. Out of any of you who have done
Like,
one
I know I have to
it's
put
a
it
lot of
down,
time.
bro. Trying to find a way around.
Researchers then wanted to really say, well, how can I look at all the genes all at once and really get a snapshot of what's going on? So this brings us to two methods one is called microarrays, which is an older method. The other is called RNA sequencing.
I touched it right. I touched it.
They're both based on the idea of taking all of your RNA from a
I
sample,
don't even want that. I don't
converting
even
it to
like
cDNA,
those.
and trying to come up with a way
You
of
want
counting how
something
many
I won't eat?
cDNAs are present for
Viruses.
each gene in the gene. Right?
I'm giving you the
Now,
method. You're welcome.
And maybe we'll go
I'll
through this
really
example, and then
do
let's
this.
go through this kind of setup, and then we'll go through the technology in the next level.
Not that I hate them,
So-
I just don't want it.
And this is just more getting at why and what kind of
But
information.
if you
have.
really want it,
So
I'll
imagine
change my
that
mind and start eating them.
This circle represents all the genes that are
It's
expressed
fucked up.
under a certain condition. Okay, let's call it a
Put it
healthy
down,
condition.
nigga. Put it
And
down,
then
nigga.
the yellow sample here is all the genes that are expressed in a disease condition. Okay? So what can we do to ask how different the disease is from
She's a six.
the non-disease condition? Well, we compare which genes are expressed in
Or anything
the
that added up.
non-disease condition versus the disease. And we look at the overlap. Okay?
So you go down.
So
Shit.
if there's not much difference between the two conditions, okay, this green area where they overlap, okay, will be quite large. But the more different our two conditions, the more genes we will see that are unique to either.
So what's,
the
wait, does
one,
it have to be
normal
specific
condition,
on
or
the top, or can I
just
choose
two
what's on top?
And by looking, for example, at these genes that are differentially expressed, we can begin to propose hypotheses about what's different between disease and you may imagine there are some diseases where there's not much difference and other diseases
So
where there
whoever
are.
has the most
This
cards,
can also be used as a diagnostic to compare two individuals where the disease looks similar, but to see if
why
there's
should
genetic
go to jail?
So I'm going to stop the
Well,
lecture
because
here, and the
everyone's
next
going
class we'll pick
there.
up with an example where we
Doesn't,
use
looks like a
differential gene
circle
expression
cards.
to help diagnose
This
two
is
very
injera
similar.
and this is the wet. This is the stew.
Why are you acting like that was fucking not rocket science? I don't want to watch this guard of honor, bro. I don't want to watch this guard of honor. You burp, nigga. You disgusting.
No, Banda. No, you already put it down.
Wait, what could I have added up?
4 plus 1.
So why are you rushing me to put it down?
Because she didn't want you to take the yeah. That's the whole point.
So if I could,
If you play with like naked methods, it's even more fun.
yeah, take a whole deck.
Alright, bro. Put the.
She already took
all
it.
of it is.
All of it. Bushy. Yeah. That's what you get. Karma's a bitch.
Idiot.
Karma's
You
a
have a 9 and a 1 here. You have a 5 and a 4 here.
Karma's a bitch, and you don't have cards now.
4 plus that's 9.
You don't have to do them in the
Yeah,
ages.
she could have done this, or she could have done this.
Her turn's not over, bro. Go ahead. You can still. It's still her turn. It's still her turn.
She already put it down. That's the whole point.
Look at this cheater, bro.
It's not cheating, dude.
Scamming Africans. What's with y'all on scamming, bro?
Oh wait, no,
Oh,
we
you
already put it
You
down.
should have put it down, bro.
Okay,
You,
but I
you
looked
know,
over.
you have already
But
put
no
it
one
down.
told me about it.
No one told
No
me.
one,
No,
because
Nick,
he's
someone said
I
so.
put it
because
down
that
already.
he just started playing. This is first time, so we don't even be like that. You already know. How is that? That is not.
Can
Pick a
you
card.
go?
Can you
Jesus
go?
Christ. Dude, that.
Let me fucking think about it. I want to take your card so bad.
You're a hater.
You're so losing. Push Just pick something up. Fuck.
That's it. That's just a face card.
Oh god, I'm just gonna let y'all tell me, bro, at this point. Oh shit. I win? Well, I would have taken it anyway, I won. Okay. about to count your cars, nigga. Don't have shit. No, don't. I don't trust you. Count it. There she go. 17. How much do you have though? Where's that? Let me feel like that. A lot less, right? A lot less, right? A lot less. Very much.
So before we start, does anybody have any questions? Anything that needs addressing? Homework, good question. Homework 10 is, homework 11 is posted, so it's waiting for you. I know, you just finished 10. 11 is there. And I think homework 9 is going to be in your hands pretty soon, graded. Let's see. I don't have the date of the final at my fingertips. Does anybody know what it is? Anybody? It's anyway. It's what it's the one. It's the date that the registrar has posted on the registrar website, so you can check that. Before the final comes, we will have a set of warm-up exercises just to help you remember the material we've been covering. We'll talk about that more as time goes on. Any questions at all about anything?
Like, what the fuck? Oh yeah, we play Will Team Curry, bro. That's a lot.
All right, well, let's get started. We've got a bunch of interesting stuff to talk about today. So today's one of those days when a bunch of things come together. In a lot of the work that you've all been doing to get to this point comes together in what I think is a really lovely way. And
this is all
we can really do something quite exciting, which I hope you will find as exciting as I do today. So I think the place to start when talking about our subject for today is going to be to go back to this. Does anybody know what this is? Anybody recognize that? I wouldn't expect you to. But let me put it this way. Maybe this rings a bell. Let's go back to day 1, week 1. We met this guy, greatest mathematician since antiquity, Carl Gauss. You remember the story? Gauss is about 19 years old. Astronomy was the hot subject at the time. It was the cool thing to be doing. And Piazzi had sighted this dwarf planet Ceres, tracked it across the sky. The planet goes behind the sun. They had not made enough measurements yet so that they could construct the orbit of the planet. It's behind the sun, it's lost. Astronomers have basically had to give up and assume that they're never going to be able to find this again. Gauss takes the measurements, the small amount of measurements that were available to him, goes off, does something apparently magical, and tells Olbers where to look when Ceres comes out from behind the sun, and lo and behold, that's where it was. So what did he do? He developed the method that we're going to learn today. He developed it when he was 19 years old. He didn't even bother publishing it for 4 years. Okay? And it's now a foundation of how we think about statistics and machine learning. It's this method called least squares. So we can start to understand a little bit better now. We built up more foundation from where we were in week 1, and we can start to understand a little bit better now what Gauss had to do, what the challenge was that Gauss took on. The starting point was to understand that there is a model for what a celestial object does when it moves around. It travels, if it's in orbit, it travels around on an ellipse. This was, Kepler discovered this, and then Newton came up with a mathematical and physical framework that predicted this.
No, I was going to figure it out. I didn't
what
put
did
it down
Gauss
yet.
need to do?
I was going to put it down.
Whoops, sorry. What Gauss needed to do was find the coefficients of this equation. This is the equation of an ellipse. 6 coefficients. Find those 6 coefficients, and you know exactly where Ceres will be at any point in time. Now, he had some measurements. What are the measurements? Astronomers looking through telescopes make measurements of where the planet is at certain times. These are the values x1 and x2. Gauss had to find the 6 values that are the coefficients of the ellipse.
This is all this is. Who's good or bad?
Well, we now understand this is a linear system.
Obviously,
This is a linear system. Why? Because we have a bunch of measurements. These are all numbers that we've measured. And we have 6 unknowns. And the equation of an ellipse in the coefficients is linear. Each coefficient is only raised to the first power. So it's a linear system. So all we have to do is solve the linear system, right? Right? All we have to do is solve the linear system? Well, let's think about what happens. According to Newton, this is a consistent linear system. Newton's the person who predicted that this is the exact equation for the ellipse. So all we need is 6 values, right? All we need are 6 equations to solve for the 6 unknowns. What could go wrong?
But did you get it from?
Oh, no.
Easy peasy. What could go wrong? Obviously, there's measurement errors.
I haven't been to Trinidad before that since I was like.
We haven't measured these x's perfectly.
I mean, here's Olbers looking through his handmade telescope, trying to estimate the direction of an object that he sees in the night sky. We have not necessarily measured these things perfectly. Undoubtedly, we've not measured these things perfectly. If we just took 6 measurements and solved this system, what would we get? We would get incorrect coefficients due to measurement error. And I can tell you that if you did this, the coefficients would be so wrong that you would not be able to find series. So what was it that Gauss did that allowed him to tell Olbers exactly where to look? The exact right set of coefficients. Gauss worked with an inconsistent system.
No.
He worked with an inconsistent system, and now I want to explain to you why it's better to work with an inconsistent system
to solve
than a
some
consistent
problems.
So
We've been
So let's think about the measurement problem. Each time you take a measurement of series, where series is in the sky, you get another equation to add to our system.
to give us the
Because each
framework we
set of
need.
measurements, x1, x2, gives you an equation in the 6 unknowns that are the A
So linear models
coefficients.
are probably
Now, if we just use 6 measurements, we'd certainly get incorrect coefficients. A better idea is to try to use all the measurements we have available.
throughout not just natural
We're going
science.
to try to
but
use all the measurements we have
mathematics,
available
physics,
and to somehow get a
especially
better
the social sciences,
estimate of these
economics,
coefficients
psychology,
than if we had
so
just
forth.
used the 6
Heavy users
that our theory tells
So
us
what
would
we'll
have been
do
enough if we had been
is to
perfectly
talk about
accurate.
linear models. And to
We're going to try to
talking
cancel out errors. We're going to try to find a way to use a whole collection of measurements such that if the measurement errors aren't related to each other, they'll cancel out.
Francis Galton was
Oh, it's going this
one of
way.
the sort of
So
brilliant
let's think
folks
about this now as a linear system, okay?
1800s, 19th
And,
century. He was a cousin of Charles
The
Darwin.
measurements
hung out in
are actually the
House.
matrix.
No, it didn't go
Spent a lot of time talking about
And the unknowns are the vector
made
A.
huge contributions across all kinds of science.
And B, in this case, is,
started the idea
well,
of,
it could be the vector of zeros because these equations have zeros on the right-hand side. So x is n by 6. Every time we take another measurement, we get another row of
one of
x.
those sort of gentleman explorers. And one of the things that he did,
Now, due to measurement errors, we can't expect that B is actually going to lie in the column space of x.
this sort of
Let's think about what's happening. X is getting, as we measure, x is getting taller and taller and taller, right? So B is also getting to be a larger and larger vector, more and more components.
thinking about how
But we've only got
what
6
Darwin was saying
columns.
about heredity.
Only 6 columns. So we're now starting to move. The solution is in a high-dimensional space.
So one of the things that
But we
Dalton
only have
did
6 columns in that space.
how
So we're spanning
variable
a
is
low-dimensional
human
portion of that
traits,
space.
like, for
So it's
example,
very
height.
unlikely that B is going to
So we
fall
looked
into the
at
column space of x.
The heights of parents and compared them to their children.
So B is not going to lie in the column space of x. Why? Because of measurement
Famous for
errors.
this chart. This is a chart that shows
The system has no
average height
solutions.
of the parents.
So what do we do?
And how far they are above the mean.
Up to this point in the course,
average height of all people.
We've talked about linear systems. They can have one solution. We learned right away how to solve that. They can have multiple solutions. We learned right away how to solve that. Systems can have no solutions. We just set that aside.
How much of the parents were above the mean.
Today,
compared them to those parents.
we'll stop setting that situation
We found
aside.
that invariably.
What happens when you have no solutions? What are you going to do? You're going to give up? Say, nah, series is gone. I'm not going to be able to do anything about this.
The heights of children
Let's
on
go back
average
to
is
being a
also
19-year-old in Germany.
The heights of children on average tend to be.
No, you're not going to give up if you're Gauss.
Is the heights of the
You're
parents.
going to say,
So he called this.
is there
this
a
called
good enough
regression to the
solution?
mean. We talk about this. Regression to the mean is a
There's no
well-understood,
solution, but is there one that's
commonly
close?
Commonly used
Can I find
idea
one
in,
that is close?
In statistics and data science, it happens
The
whenever you
measurements
have a
include errors,
random
but that does not make the
phenomenon.
measurements worthless.
We look at the same example, and then
We just need the principle
more
approach
random events have happened. What happens
to do the best job we can, given the errors that
So
are
it's seen in baseball season.
Let's see how we can do that. That's what we're going to do today. That's our job for today.
First place in each division at the All-Star break.
So let's think about this mathematically. We understand.
They will regress to the mean.
Now we're back to
The influence of random
writing it as
events.
ax equals b. It's the same equation, but let's write it this way. We
Examples
now understand,
will-
at this point in the course, we've built up enough understanding
And this is actually
that if
what
we have an m by n a,
Galton coined this term.
and there's no solutions to the
And we now
system,
use it not
the
just
reason for that
this notion
is that the
of
columns of A
we use it
don't span RM,
anytime,
and B is not in the column
anytime
space.
We're making a
There's
prediction
space
about a numerical
in RM that's not spanned by the columns, and B doesn't
Now
live
really
in the
refers
column
to,
space. B lives in the part of RM that's not in the column space.
Any kind of prediction about a numerical quantity.
So here's a good example.
So,
Like, we've
regression
looked
is
at
our
this
subject
example
today.
many times. Here's a particular
Regression is our
subspace.
subject. We're gonna talk
It's a
about
column space of some
how to
matrix.
build a model
This green plane.
to
And here's B
summarize
floating
data.
out here, and it's not in the green plane. And that is
Now, a model is
a good example
necessarily a
of
concise, smaller, distinct
inconsistent
data.
system. B is not in the column space of A.
And typically, the models that we use in, for example, in statistics,
Now,
oftentimes
the
are
point is
equations.
that for the problem of series' orbit, the reason that B doesn't lie in the column space of A is because we've measured B
anytime we have
with error. And
some
we've
kind
measured,
of
in
process
fact,
that has,
even the column space of A is
that has
not quite
values
correct because of our
associated with parameters
measurements.
associated with it, and we use it to predict a number, then we are building a
So,
regression model. So
what are we
regression
gonna do?
is constructing an equation
We're going to find a good
that describes
approximate
relationships
solution.
among variables. So let's look at
There's no exact solution.
regression.
There can't be. B is not in the column space
So
of A.
imagine
There's no
that I
solution
have,
to the system
a
that's
bunch of
that's
data.
exact. But is there an approximate
like
solution?
this. And
Well,
I notice after plotting that,
if we make the assumption that the
they
measurement errors are small,
appear to lie more or less on the line.
then we should be able to find an x
Heights of the children compared to the parents.
such that although
You notice
AX
they were
doesn't
approximately
equal
lying on a
B,
line. We could
AX
decide
could be
model
close
this
to
data
B.
Watch how she looked her hair.
as if it were a line. We could look at this data.
This is actually the whole key idea. The whole key idea of the whole lecture today is what I just said.
Here.
AX is not going to equal B,
I should model these.
but maybe we can get AX to be close to B.
Or I look at these points and I say, I sort of see something that looks like a log curve here.
In fact, we'd like it to be as close as
Maybe I can
possible,
model
right?
these points using a logarithm function.
We'd like this to have a property where we're going to find the x that's going to make AX to be as close as possible to B.
Exactly lie. on the curves that we're thinking about.
We're looking for an x such that when we multiply it by a, we get something that's very close to
In
b.
this case, they don't lie perfectly on a line. They're just close.
Here it is.
And these points don't lie.
Here's b again, and here's the column space of a, and we're looking for some x
So
Ugly,
it's
bro.
not
such that when we use it to form a combination of the columns of a, of course we get a point in the column space of a, and that point in the column space of a is as good as we can get
How do we choose
towards b.
How should we find the best linear function or quadratic or whatever?
So we can actually think of, in some sense, the error of the approximation of ax to b as the distance from
So
ax to
this
b.
is the subject
I'm
of.
going to expand on this in a minute. This is a fundamental principle. But remember that when we talk about the distance between two vectors, it's the norm of their difference, right? That's the distance between two vectors.
We should recognize
So this is the distance, the geometric distance, if you measured with a tape measure, the distance between this point and this point. The distance between this point and this point.
So we'll talk about a collection of variables or a collection of measurements. We'll put them in two categories. We'll say that some of the
So
values
now we can start to
we'll refer
formally express what it is we're looking for. And this is called the general least squares
and the other
problem.
ones, we'll refer to
We're looking
as
for a
dependent.
good solution to an inconsistent system.
Now,
It's not a
why
perfect
we put
solution.
some
There
in
is no
one
perfect
category and some in the
solution.
other will become
But we're looking for the
clear
best possible,
over the course of the
the
lecture.
nearest, the most nearly close
Now,
solution, the best
basic
alternative.
task we have, when we wanted to
The least squares problem, the general least squares problem, is to find an
given
x
some set of independent
that
variables
makes the distance
and some set of
from a to b.
dependent
Because
variables.
you cheated.
And our job
Now, why is this called least squares?
parameters of a model.
Why is it
No,
called
Let's say
this
we
cheating
decide we're
again.
gonna use a line.
This is the distance. This is the geometric distance in space between ax and b.
For example, take
Why is it called
slope
least squares?
as the parameters of the line. But we'll use
It's because if you
slope and intercept as the parameters.
If you look at this expression, the squared
We want
distance,
to estimate the parameters of the model that describe
then
how the
let's
dependent
call
variables
ax y. Let's just give it the name y.
Bro.
Then the squared distance between ax and b is the squared distance between y and b.
into our model.
But let's write this expression out.
Apologize.
What is this expression? It's each component of y minus the corresponding component of b.
The independent variables.
Square that quantity and add it up.
Bro.
Now, what we're going to do
This is in statistics called the sum squared
is
error.
we're going to model, we're going to treat this problem in an algebraic.
Think of y as an estimate
algebra,
of
obviously.
b, and b is the thing we're trying to estimate. Then yi minus bi is the error for that component, and all we're doing is squaring those errors and summing them up.
Shit.
Info matrix X.
But it's the same thing as the squared distance, the geometric distance in space. This is a crucial idea. Just tying geometry to statistics. The distance in space between these 2 points is the sum of squared error.
Designed a particular
Square
sum
God,
structure,
this is
design
like
matrix.
ugly.
The dependent variables we call
So this means that we can, when we want to solve this statistical problem of minimizing error, we can think about it purely geometrically.
Y.
Shit.
Think about it purely as just moving around in space and finding the closest point.
Might use. The parameters are always collected into a vector called B.
Let's see how to do that. We want to minimize the length of x minus b. We want to find x such that it gives us a point in this space that's as close as possible to b.
Traditional notation of statistics. So we have to sort of get used to this way of
And that's the same as minimizing the sum of squared error.
vectors and matrices that work.
All right? Geometry, statistics, machine learning, all in one concept, tying them together.
See, there's all three.
Okay, so we've got a situation.
Let's make this a little bit more formal. We will say, if we've got some A, some matrix
We want to
A,
think about what is the
and
best
some B,
line we can
that's
draw
in
in this table.
the codomain of A, so it's in the same
So we're
space
going to
as
start with the simplest
columns of
possible
A.
model. Simplest possible linear
Then,
model.
what we would say a least square solution is
Parameters, a slope and an
of ax
intercept.
equals b. Now, there is no solution of ax equals b. This is a system that probably doesn't have a solution, but a least square solution is something different. A least square solution is some x hat such that the distance from ax hat to b is smaller than it is for any other possible point we could have chosen. In other words, ax is closer to b
Useful because
than any
so
other
much
point
data
in the column space of A.
tends to follow a linear relationship between
It's a least square solution.
independent and dependent values. So just to make clear,
And we also write this more commonly. You'll see it more often written this way. Okay. So we're saying we want to, this notation says we want to minimize this
So we
quantity
have to ask this question.
by varying x. Okay. So the minimum over all possible x's
How should
of
we
this
fit this
quantity.
equation of the line to the data?
And the art part says, tell me which x this is. That's what I'm looking for. So argmin of x says, give me the x that minimizes this quantity.
It's as close to
And you will see this kind of expression for the rest of your careers in computer science, data science, machine learning, as the way that we talk about minimizing error, minimizing function,
the picture of what
training,
we're talking about.
training models.
Suppose we
All
have
of
this
these
line.
are expressed using this kind of notation, where we're minimizing a function, and we're looking for the argument x that minimizes that function.
To the line by saying, what if we
So this is a problem. This kind of problem is called optimization.
distance from the point to the
Optimization.
corresponding line
And today we're going to solve this optimization problem.
So what are we saying? We're saying
So
what we're interested in is how far the points are from the line.
Let's think about this least
The
squares
line
problem.
we're going to move, right? We're going to move the line around.
one thing to keep in mind.
in
Sorry.
measuring these distances until we found a way to make the line as
Good.
close as
Okay.
possible.
One thing to keep in mind is that no matter what x we pick, ax is going to be in the column space of A.
That
Right? Because what is ax if nothing more than a linear combination of the columns of A?
vertical line. We say,
So,
I'm interested
thinking
in,
about a problem,
this data point.
we've got a
xj,
situation
yj.
where B is outside the column space of
Independent
A.
value is xj.
We're looking
And
for an
if
x
I had
hat
used the line
such that it represents the coefficients we're going to use to combine the columns of A to get as close to B as possible. So imagine moving around inside this green plane,
That's this point.
moving around until you get as close to B as
We're
possible.
interested in the difference between, or the distance really.
X
between
hat
this
is the
point.
combination of the columns of A that gets us to this point.
So we use some terminology for this. We talk about this
Now,
distance.
the point is that for this to be a least squares
The
solution,
residual is what's left over.
what that means
when we use
is
the
that this
line.
point, this ax hat point, is closer to b than any other point in the green
Data point.
plane.
There's some corresponding point on the line that has the same
Any other x that we had
coordinate.
chosen, giving us some other point like one of these red points,
The same x coordinate, but we add
would
one
give us
no,
a point in the green
line
plane further
or the y
from b than ax hat.
You casting a spell?
Is that clear? Oops. This residual is the thing I was talking about.
So?
being
Hey,
how close the line is to the point.
That's our problem. That's the problem we need to solve today.
So we call yj the observed value.
We've got 45 minutes to do this. Let's see if we
The
can.
line would give us beta
You
0 plus
watched
beta
this.
minus j.
We're gonna need a little more theory, just a little bit.
Spell.
And the difference between
You're almost there. What we built up to this point is almost everything you need to know except for two facts
Sure.
that we need to now spend
we
a
still
couple
haven't said
minutes on.
exactly
So that
how
we've
we're
established
going
what
to
we
talk
need to
about
know
this
to solve this
distance between the line
problem.
and
So we need to bring in just a little bit more theory. We need to extend the ideas that we had in the last
Let's,
lecture. So the last lecture, you remember we talked a lot about orthogonality, perpendicularity.
So,
and projection.
in fact, we don't do that.
So we're going to talk about a little bit more about those two things.
Lengths of these things.
And basically, we just need to take some of the ideas from that lecture and generalize them to arbitrary subspaces.
But one of the reasons.
So we need two theorems. One is called orthogonal decomposition,
Positive and some are negative.
and one's called best approximation.
So
So let's look at these two theorems. They're both quite straightforward.
The negative residual. We don't want to
Let's first talk about orthogonal
add up all
decomposition.
residuals because the positives can cancel out the negatives.
Watch that.
Here's the statement.
So one reason.
Let's
say we have a subspace.
To make everything positive.
And it's a subspace of Rn, and then we've got some other point y in Rn.
Important to the same extent as distance.
Then it's always the case that we can write y as the sum of
So
two
we're going to
things.
square these residuals
A vector that's in
and sum
the
them up. And
subspace,
that's going to be the
and a
measure
vector that's orthogonal to the subspace.
Eyes,
for
bro.
asking how close the line is to the points.
No, we actually looked at this, yes. We looked at
This
this
squares line.
on Tuesday, okay? You recall we were looking at pictures like this, and we saw
Wait, wait,
that
wait.
we think of this subspace, we think this line as a subspace, then we can always take some point y,
Wait, that's a
decompose it into two
nine.
things.
These
Y
coefficients
hat,
are then called the
which is
regression
in the
coefficients.
subspace, and y minus y hat, which is orthogonal.
So this is the problem we're trying to solve today.
We can always do
Find a line.
it. It's pretty obvious that we can always do this for a line.
Now we have a precise definition.
But all we need to do is establish that we can do it for an arbitrary subspace, and here's what the situation looks like. Here's an arbitrary subspace, and here's some point not in the subspace, and we have to be able to say that we can always do this
All right.
no matter, it doesn't have
It's
to be
called a
one
least squares
dimensional like it
line.
was
So
in the
let's
last.
ask ourselves now.
So this is actually straightforward. I'm not going to prove it to you, but I want you to sort of convince yourself that this seems quite possible. That anytime we have some point y, and we have some subspace, we can say, well, y is just the sum of a point in the subspace
Then it would
and
be
a
the case
vector
that
that's
each
orthogonal.
prediction would be equal to
Okay,
the
it's just the ice skaters' shadows being projected down onto the ice surface.
X1 would equal Y1.
And this theorem simply says we can always do that.
That's if it so happens that the line was
And-
inconsistent every data
So
point.
I have time.
Just like we did in last lecture, we'll say that y hat is the orthogonal projection of
What
y
is this?
onto
It's
the
nothing
subspace
more- it's nothing
W.
more than a system
And this is the way we denote it.
God will win.
We say projection onto W
we're
of
looking
y.
at since day one. This is a
Okay.
system of linear.
So that's pretty straightforward.
What are the knowns?
The orthogonal decomposition theorem says no matter what subspace we've got, we can always take a point and break it into two things, a point in the subspace and a vector that's orthogonal.
So let's write this in our familiar matrix vector way.
Now, the other theorem is a little bit more
As follows.
involved,
The knowns.
and it's called best approximation.
And here's the other knowns, right?
We actually kind
So
of
this is
touched on
just
this
the same
on
as
Tuesday, but I kind of glossed over it. Now we're going to treat it very seriously, head on, and we're going to be formal about it.
The matrix X,
And here's the statement of the theorem.
notice.
Again, we have a subspace W,
That we're
and
doing.
again, we have a vector Y.
Because we put a
That's
column of
not
ones into these
necessarily in W. And now we have y hat, the orthogonal projection of y on W. So
Each one of these
we're
equations
using that best approximation,
has
or
the
that
V0
decomposition
in it.
theorem we just
By
stated,
putting
which is
this
that
column
for every y, there's a y hat.
and
That is the
creating
projection of
a
y on that
vector
W.
as beta 0 and beta 1. You see that
If we
beta
have
0
that
is
projection,
multiplied
then
by
the theorem
1.
says y hat is the closest point
Play the role of these
in
beta
W to
0
1.
equations above. So this system, x beta equals y, is exactly the same as these equations. Just taking into account the fact that we've added a column of ones so that we can have this intercept value, this beta 0,
Really?
constant. It doesn't depend on
What are
the
you
x's.
doing?
Does everybody see how that works? Okay, so this is what the linear system would look like if it happened that we could draw a line through every point exactly. Now, of course, the whole point is that the data points don't actually lie on the line. There's no parameters beta 0 and beta 1 for which this is a consistent system. There's no solution to this system, right? It's an inconsistent system. There's no parameters for which the predicted y values in x beta are equal to the observed y values in y. So x beta equals y has no solution. So we know that it's an inconsistent system. Now, we've decided that since the data doesn't fall on a line, we're going to use this criterion to choose the betas and the beta 0 and the beta 1. And what is this criterion? It's nothing more than beta 0 plus beta 1 xi, that's the predicted y. That's the prediction for y. And this is the real y, the observed y. So what is this? This is simply the sum of squared error. Right? This is the error in the prediction. We're squaring it, and then we're summing over every data point. So this is just the sum of squared error. What we saw in the last lecture,
I want to go to sleep fully
that
today.
this kind of expression, sum of squared error, we can think of geometrically. Because this is just another way of writing this. This is just another way of writing this, right? This is x beta, y. We're taking each
Smell
component,
sulfur,
squaring
Asia,
it,
'cause
summing
I
it.
smell sulfur.
That's exactly the norm
You smell sulfur?
of the vector x beta minus the vector y squared.
I smell sulfur.
But, what is the norm of x beta minus y geometrically? Now think, what is the norm of the difference of two vectors geometrically?
So, you know, just want to
It's the distance between the two vectors, right?
And you got to choose this and final.
If we visualize these in space, we would say this is the distance between the vector y and the vector x beta. It's the distance squared. But
Those
it's-
are not his teeth. Those are like.
So the sum of squares of residuals in our linear
those
model,
are-
the sum of squares of residuals in our linear model is exactly the square of the distance between x beta and y.
Oh, Asia one again?
Okay, I hope you're seeing now that we're about to connect to Thursday's lecture, okay?
Kind of Jack. Take my
Because
cards.
what did we find out
God prevail.
about solving these types of problems?
What'd you get?
What is x beta? X beta is a point in the column space of x. So we're looking for the closest point in the column space of x to y.
What are you doing? Put that down.
This is
Work with
a
Asia.
least squares problem, just as we've defined it in Thursday's lecture.
No, what? No, I'm not
And so
sure.
here is the key idea for today. Finding
Now you do get.
the best linear model is equivalent to finding the least square solution of x beta
Like,
minus
that's
equals
so
y.
funny.
That was not
The least
gonna.
squares
Yes,
solution
God prevail.
that gives us the best linear
Fail?
model.
Bro, whatever.
No, isn't this the last one?
That's the last one.
That gives us the best linear model.
I ate, so I would have picked it up.
And it's because the least squares criterion is about the distance between points in space.
I didn't win, but
And the
I got
criterion that we
closer
adopt
to-
for choosing the best linear model is the same thing. It's
Let
about
me
the distance between x beta and y.
I don't need you to count it. I'll count it myself. Thank you very much. Jeez.
So, with that, let's do a question.
14. That was so close to being 13. If she would have counted, it would have been 13, the devil number.
13 the double number?
Yes, the evil number.
I
Yes.
thought it was just 6.
No, 13 is a double number too. Why do you think it's Friday the
Isn't that
13th?
why there's
the
not
Friday
like,
the 13th
a
is the-
13th floor in like most places?
The 13th floor
I
of
don't know,
West
but
Campus?
the Friday the 13th is supposed to be evil.
There's
That's
13th
like,
floor West Campus?
who won? Who won?
You talk too much.
You know.
You know. So much.
Oh, because there's a small
so much.
mistake.
So much.
Which did lead to you getting a lot of cards, so.
So much devilish talk.
No, it really didn't.
Yes,
It did. It did.
Because
Helped you for the whole
what you got
game.
after
Everything
was,
else you got after you came from the chats.
Yeah.
The chats you stole.
Regardless, he would not have picked up that card, bro.
Yeah, but
Yo,
like,
you should be a freaking storyteller. Like you're fucking dramatic as hell.
yeah.
Why is it a semicircle, bro? Make it a full circle. Everyone's invited. I feel some type of way before even drawing it right now.
Dude, I'm just putting 4 cards in the middle. Chill out.
Okay,
I'll just go.
Asia's turn. It's Asia's turn.
Calm down.
Asia Warden's turn.
I have to spell my name down.
Asia Lee Warden.
Down. Okay, we'll go this
Oh,
way.
this way?
I guess
Yeah.
so.
I guess.
Go, Sada.
Damn. What the fuck, bro? Bro was really like looking.
You know, making sure her fucking quick hands don't move fast.
There's a lot of cards right here.
No, show us. Go show us. Hey,
Go, Nicholas, you know, hurry.
it's my turn.
What did you get?
The king.
All that, I don't cheat. That's why I pride myself in not cheating. Unlike some people.
What about on exams?
Oh.
Ooh,
Hey.
fuck that.
Hey, hey, let's not be hasty, guys. Why is your hand in there?
Go, Sada.
Dude, it's literally over
Ew,
here.
why
bro.
is it on that?
Sada, it's your turn.
Ew, why is it on that? You're
On my
so
stomach.
mean.
Sada, it's your turn.
It's so ill, bro. Stick it out. Have your hand on the table flat like this, visible to everyone.
Go. What?
I'm so sorry.
fart again?
What is that? Is
Twice.
that a 3? Like, it's not like, okay.
It's a 3. Where did they come from?
No, I thought it was like, I saw 3 plus 3, so maybe it was like, I don't know if you flipped that. I don't know. I'm just talking.
Are you trying to swindle her? Is that what I'm watching right now?
Shut up.
So,
Hey.
for what, bro? Shut up.
It just feels some type of wave if you're doing legally now. What else are you gonna steal from all of us now? You're gonna say 3 plus 3 equals something, bro? You're gonna
3
find
times 3 is
no 3 times 3 equals 9, bro? Is that what's gonna be going on now?
That's a completely different card.
Just put it down. Use your book.
What was my name? What the fuck?
Oops.
Oh my god, it's an Ethiopian thing.
Wait.
It's an
4,
Ethiopian
5,
thing.
6, 7, 8.
Oh my god, it's an Ethiopian thing.
How did you get 8? It's 4 plus 3. 4 plus 3 is 7.
Oh, I don't.
Now tell me now, is it Ethiopian thing or African thing?
No, it's not.
Hey, don't disassociate yourself if you're both cheating now.
I'm white.
Can I go?
You're not one
Just
of
in
her?
case, yeah,
No, it's next
okay.
to her.
No, it's not my turn, right? It's
Oh,
not her turn?
Yes.
See, I'm not cheating like you, I'll be honest. I'll be honest, you would've took it. You would've took it if it was yours. It wasn't yours.
You know, if you had cards to steal, I would steal them. But you don't have any cards to steal. Let me see.
Nigga.
7. Stop! You can't cover your.
Who
cards.
was hiding it? I just ended my arm on it. I have it on your side, look at it too.
Just put it right here so everyone
Shit.
can see, bro.
Everyone, y'all, 7. Look, 77. Look, it looks like the substitution.
6, 7.
Go show everyone. Show it and show these face to have. Hey. This feels fraudulent.
How is it?
Add it. Show us. Show us. What plus what equals what?
6, 4 plus 2 is 6.
Okay.
How am I being fraudulent, bro? Like.
Spurs fan that goes with you. Shit, man. Ooh.
Bro, I want that 7 so fucking bad. Come on. Come to me. No, I'm sorry.
Devil magic. That's what she's
I didn't
doing right
even-
now.
I didn't even want the 3, bro. I wanted that 7.
So what do you sign away to? Was it Tottenham's this misfortune for all these years? Is that what you're winning right now?
Why are you showing me?
Well, I'm making sure everyone knows I'm not a cheater.
I'm not a cheater either. You covered up your 7.
Huh?
I said you covered up your 7.
Okay, I'm not cheating though. I'm supposed to cover up my 7.
Who was your son?
Don't get a 3 solely.
I got a 3.
Hey!
How?
Hey,
No,
9
no,
minus.
No one can take your 3s now.
Yeah.
9 minus.
Dude, it's 9 and 9.
9 minus 6 equals 3, bro.
Shut up. That's 4.
Okay. I believe you.
I had a 10 earlier.
No, you had it earlier, bro. You put it down already. She put it down already.
Okay, I know. I was just checking to see if this stack was good at the time, so no one can steal like this. Because then no one can.
Show us.
Show and tell.
God, bro.
Something feels suspect right now, bro. Why? No. Suspect.
Bro, there's literally nothing suspect.
If she wins, I'm going upstairs because this room has been lost to Satan and his works. What is that? What is it? Did I say a 7?
No.
Do you have any J's?
Yeah, I think I do.
God has forsaken this room. I will leave after this game. Hey. 3 + 3 = 6. Is that how it works?
No one can steal her threes. She swapped it with the threes.
Yeah, she has 4 threes.
So, rally or something. Take her jack, something. Does anyone else have a jack?
That's bro.
Are there jacks on the... Oh, shit.
Why do you look like that, bro?
God has forsaken this room.
Genuinely, what do you...
I'm telling you, God has forsaken this room. I'm leaving. I suggest y'all do it too. I suggest you go upstairs and you go home, bro, because God has forsaken this room.
Damn it, I should have stole your 7, bro. How are you getting
Fucking eyes might pop out of your eye socket, nigga.
Can you flip it for me? I don't want to touch anything in here, bro. Take that one. Hey, Chai, put it in the middle.
Hopefully you don't get anything.
Take 7.
No, she does.
Sit down, come here.
No. I don't want anything to do with this devilish game anymore.
Let's play BS.
Hey.
Come here.
Or blackjack.
So a game where she can legally lie? Hey.
Damn, Damn.
Come here.
I don't trust that.
Come here. I said sit down.
I want to drink.
Sit down.
I want to drink.
Sit down.
I want to drink.
Okay.
Why are you getting
You leave, you don't get any genetics help.
I can't get a drink.
Nope.
With what money?
I don't even have an ID, bro. I had to go upstairs.
Wait, where did the recording go?
Okay, should we play BS? Or should we play blackjack?
You've been playing BS this whole time, sister.
that was clever.
Hey.
BS or blackjack?
BS.
Don't fucking touch my shit, Nick.
I don't even want to touch anything regarding your money, bro. That might be stolen. Too. I have no idea where your funds come from.
No, like you give.
Is this Palm? Oh, this is Tylenol. I actually isn't gonna get a drink. So can you work on my cards for me? I don't want to touch it. And you can bless this water or something. Bone dried. But see?
Who has ace of spades?
I'm not touching that. I'm not touching that until there's